Okay, hello everybody. Good morning, good afternoon, wherever you're coming in from. Great to see so many people tuning in. We'll give things a few minutes here. Great to see so many people coming in, a lot of people who are regulars to our program, and a few new ones too. Welcome to Club EvMed. I'm Charlie Nunn. I'm at Duke University in the Department of Evolutionary Anthropology and Duke Global Health, and I run the Triangle Center for Evolutionary Medicine. And TRICEM, as we call the Triangle Center for Evolutionary Medicine, is the organizer of Club EvMed. But we do this with a bunch of other evolutionary medicine centers and the International Society. So I'm going to paste that in here. And I'm also going to paste in some other information for everybody. A lot of you know this, I hope. We also work with the International Society for Evolution Medicine and Public Health. And I always like to call attention to the society's journal, Evolution, Medicine, and Public Health. And you have a link there. And there are two special issues you should be aware of. One on microbial evolution that launched pretty recently, and another one on eco-evo applications in cancer. And that one's closing pretty soon. And very importantly, I want to let you know about our conference, which will be in Kiel. The registration is open. The abstracts have closed, but there's usually an opportunity for late-breaking abstracts if anybody missed that deadline or something exciting comes up and you want to present at the meeting. So anyhow, thank you guys very much for tuning in. And it's a real pleasure today to introduce India Schneider-Kreis as our speaker. India was nominated by one of our partner organizations, and of course that's the Center for Evolutionary Medicine at ASU. We always ask our organizations who co-lead this to put in nominations, and this year Ann Stone nominated India as a member of the Center for Evolutionary Medicine to highlight the work that's going on there. Ann couldn't be here today, so I have the pleasure of introducing India. And that's actually pretty easy to do because India was a PhD student in my lab. I got her PhD in 2017 in the evolutionary anthropology program here at Duke. And she studied a very enigmatic and very charismatic and intriguing species, the gelada baboon. Oh, wait, am I supposed to call it the gelada or the gelada baboon? What was that?
Gelada.
Gelada, thank you.
Gelada.
Oh,
Good
okay,
catch.
it's been too long. It's been too long. She graduated in 2017. I've already forgotten everything she's trained me to do in terms of terminology about the gelada. But a very fascinating species, and as you're going to see today. After her PhD, she went on to two different postdocs. The first one supported by an NSF postdoc fellowship that was at Stony Brook University and also at University of Washington. And then she went on to the Center for Evolutionary Medicine at ASU. And they must have liked her a lot because they offered her a job. So she's been an assistant professor after that, starting in, I think it was 2022 or so. Is that right about that?
Yep.
And anyhow, India does amazing work. As you're going to hear, she's the co-director of the Simeon Mountains Gelato Research Project and also another primate project, the Zalaki Project. Hopefully, I'm pronouncing that correctly in Ethiopia. And you're going to see that her work is very strongly rooted in a One Health approach and engages with evolutionary medicine as well. And in general, she aims to understand how infectious disease shapes the ecology and evolution of host species. So, with that, India, I'll let you take it away. Welcome to Club EvMed.
Thanks, Charlie. Thank you for that introduction. And it's so great to see so many familiar names and some familiar faces for those who have their cameras on. I'm really excited to be here. I'm really excited to catch you up on what I've been working on. Um, so I'm gonna go ahead and share my screen. And then I am going to go into presenter view. No. Okay.
Looks
Did that
great.
work? Can you see my screen?
Yep, looks great.
Perfect.
Beautiful
All
picture.
right. So I will begin. So as Charlie said, my name is India. I'm an assistant professor in the School of Human Evolution and Social Change and in the School of Technology for Public Health and the Center for Evolution and Medicine, all at Arizona State University. So at ASU, I have my lab is called the Wild Worms Lab, and it's essentially become a One Health incubator lab. So we have a ton of undergraduate students, master's students, PhD students that work across disciplines on different dimensions of health and disease. And across, we have students from engineering, we have students from anthropology, we have students from biology, and from conservation biology. So our team works in the lab, in the field, and in engineering workshops. So it's been really fun, and I'm really lucky to have such an amazing group of really talented and ambitious researchers that are working with me and driving a lot of the research that I've been working on that I don't have time to do myself. So that's been really great. And the work that I'm going to be talking to you about today was actually born in at Duke when I was a PhD student in Charlie's lab, but it's taken this long to kind of come to fruition. And it's been taken to its final form by my very talented PhD student in global health, Medhavi Verma, here, the second from the left. So much like this project is an extended phenotype from my dissertation, today I'm going to be talking to you about parasite manipulation. So for those of you who are maybe not familiar with parasite manipulation, I think it's one of the coolest phenomena in the world. And it's often talked about as an extended phenotype. So parasite manipulation in that a parasite is able to systematically and stereotypically change, alter the behavior of its host. So this often takes the form of manipulation that alters the behavior. So I'll give you three kind of examples here to orient you that I think are among the coolest and the most spectacular, the most striking. So here on the left, you've got an ant that's infected with the often called the zombie ant fungus, Ophiocordyceps, which is obviously the inspiration for Pedro Pascal's The Last of Us. So you can see here, normal ants don't have a fungal stalk growing out of their necks. This one does. So this fungus, when it infects the ant, it alters the ant's behavior such that the ant leaves its nest and crawls up some vegetation and then grabs on to a vein or a part of a plant in kind of a death grip with its little mandibles as the fungus begins to grow and then essentially results in this protuberant stalk. And this stalk will then have all of the spores. And the reason it goes up and it alters its behavior is because the height that it ends up going to brings it closer to the optimum for the fungus in terms of humidity, sunlight exposure, all that stuff. So in this case, the fungus is altering the behavior of the ant towards its own dissemination. You see something similar in the middle. This is a horsehair worm coming out of a probably dead at this point, praying mantis. The horsehair worm is a nematode that infects crickets and praying mantises. For this stage of the parasite, it needs to promulgate in its next stage in water. But mantises are not aquatic. So essentially, it precipitates the suicide of that host in order for it to reach the water where it can produce its next stage. And then finally, on the right, this is one of my favorites. This is an ant infected with Dicrocelium dendriticum, which is the liver fluke. And this is just a cool life cycle in general. So it starts with basically it's in a cow, and then the sexually reproductive stage is in a cow, and then it goes into the environment. It goes through a snail that where it gets into a different stage that then exits in a slime ball, and then the ant eats the slime ball, and then that stage in the ant takes over again the neural networks of the ant and directs it to climb up grass blades and then stay on those grass blades. And that increases the likelihood that that ant and the parasite inside it will be eaten by the cow or the ruminant, thus completing that life cycle. So this has been kind of the most detailed in insects and somewhat in fish. And oh, that's the extended phenotype. So the extended phenotype meaning that the parasite is its own phenotype includes the behavior, the altered behavior, the altered locomotory patterns that it produces in the host. So this has been most detailed, most extensively detailed in insects and to some extent amphibians and fish, but it also exists in mammals. And the most famous example of this is, of course, Toxoplasma gondii, which is a protozoan. So in the toxo life cycle, the cat has the adult stage and then produces oocysts that are shed into the environment during defecation. Those are ingested by the rodent, and then the rodent host it develops into tissue cysts in the tissue or the musculature in the rodent, and those cysts cannot become the adult stage until they are consumed by the cat during predation. So, where does the manipulation come in? I'll tell you. It comes in because in the rodent, infection actually increases the predilection of the rodent to approach the feline definitive host. So when you have an infected rodent, it is attracted to, it's not just that it loses a risk aversion, but it actually is attracted to the smell of cat urine. So infected rodents are more likely to approach cats, which of course increases the likelihood that uh, that the cat will ingest the infected animal and that parasite can get to its next stage. So, in all of these, the pattern that you might be noticing is that the parasite manipulation alters the host phenotype in ways that increase the fitness of the parasite, and they often come at a great cost to the host. So, in all of these cases, the cost to the host is that the host dies. So, it's a pretty substantial cost. And it's really amazing. The general strategy of manipulation has really evolved across parasitic taxa. It's really remarkable. But there are also cases of parasite manipulation that aren't just killing the host so that it can be transmitted to the next stage. But these are much less robustly characterized, and partly because they're really hard to detect, especially in the wild, right? Kind of the big spectacular killing of the host is fairly easy to observe, especially when you have something like the horsehair worm that is like coming out physically doing an alien outside of the praying mantis. But other types of manipulation, you really need a lot of integrative and long-term data to observe these effects. So, for example, one example of this is Taenia crassyceps. Taenia crassyceps is a tapeworm, one of my favorites. And Taenia crassyceps cycles between its basic host is it goes between a fox in its adult stage. So the adult stage is basically it's what you think of when you think of a tapeworm, but it's really a series of segments in the gastrointestinal tract. Each of those are hermaphroditic and produce tens of thousands of eggs. Those are shed into the environment during defecation. They're distributed around. The rodent ingests those while it's eating, and then it grows into a cyst. That cyst cannot become an adult until it is consumed by that definitive host. So it's this exploitation again of a predator-prey cycle, just like with toxo. But unlike toxo, there is another layer that facilitates another type of manipulation because Taenia crassyceps doesn't just it's not a one-to-one you eat one egg and it becomes one cyst because it is asexually budding in its larval form. So you can have a single infective event that produces an initial cyst and then that initial cyst begins asexually budding and can produce a bunch of downstream daughter cysts. So this means that there are kind of two opportunities for the parasite to increase its fitness. you've got the facilitation of transmission, right? So the same kind of exploitation of that predator prey increase the likelihood that the prey will be eaten. But you also have this space of is there variation that can be exploited in the ability of the parasite to asexually proliferate and produce more daughter cysts? So there's a group out of UNAM in Mexico City that has been working on this system in the lab for several decades at this point. And they noticed that Taenia crassoceps preferentially established in females. So when they did in vitro experiments, they saw that treatment with estradiol it led to an increase in proliferation of those larvae. So more daughter cysts were produced, and they were produced faster. When these were treated with testosterone, the cysts died. So testosterone has a real cysticidal effect on Taenia crassoceps in its larval stage. So does this mean that Taenia crassoceps, when it encounters a male host, just kind of gives up? No, it doesn't. It finds a way. So when they did these experimental manipulations, they infected Taenia crassoceps. No, they infected the rodents with Taenia crassoceps, and what they saw was that infection increased the aromatization of testosterone to estradiol in this living host. So you had a situation where the infected individual, testosterone was essentially depleted across these individuals. Estradiol was off the charts. And this, as you might expect, led to a complete cessation of mating related behaviors, copulation, territoriality, aggression, and a complete atrophy of the reproductive organs. But this is really cool, right? And this is certainly a manipulation, but this has only been seen in a laboratory environment. And there's a lot of things in the lab that may not represent or entirely reflect what goes on in nature. There could be like something I think about a lot is we don't have a good idea of what the infection intensity was that they established in the rodent hosts. So we don't know if that intensity is something that really ever happens in the wild. So perhaps it's possible that there would be a certain threshold that you would have to reach for this conversion of testosterone to estradiol. To happen, and that perhaps that doesn't really reflect reality. So we turned to the field to look at this relate at a relationship that has a similar pattern in the wild. So this is my all-time favorite parasite, the tapeworm Taenia serialis. So this tapeworm, very similar to Taenia crassoceps, it cycles between a canid definitive host and in this case, gelada monkey, not baboon. So the geladas ingest the eggs during foraging, and those eggs develop into larvae in the musculature. And those larvae then cannot become adults until that gelada is ingested by that definitive host. And just like Taenia serialis, just like Taenia crassoceps, Taenia serialis has the capacity to, from a single infective event, turn into multiple downstream daughter cysts. So it has an asexually budding larval stage. So a little background about geladas, the best primate in the world. Mm, Geladas are not baboons, as we've talked about, but they are related to baboons, but they are the last remaining species in their genus Therapithecus. Uh, and they only live in Ethiopia, only in the highlands, and they are graminivores. So that's what gives them, they're constantly interfacing with the soil, they're coming into contact with these eggs all the time. So I'm lucky enough to have worked with for a long time and now co-direct the Simien Mountains Gelada Research Project, which has been studying a population of geladas in the Simien Mountains in northern Ethiopia for now 20 years. So it's this kind of long-term field site that is really special and really important for asking questions about diseases, about impacts on about parasite-host relationships, because you can really look at the impact of these relationships over a lifetime, over a lifespan, and over multiple generations. And one of the things that also makes this such a great species to ask these types of questions in is because of the asexually budding nature of the particular parasite of Taenia serialis, because it turns into these massive cysts that are observable and that, without knowing what it was, the research site had been collecting data on for 20 years. So this is a really unique opportunity where we were able to leverage all of these data about these protuberant cysts to ask these big questions about how it might impact geladas. This is just a fun picture of what it looks like on the inside. So within this giant, massive protuberant cyst coming off of this female's face, if you were to open that, and this one is not her face, this was a male arm, but if you were to open that, you can see that there are these like fluid-filled sacs in there, and then within those sacs are little they look like grains of rice. So each of those is a larva, and each bladder pack starts with one larva in it. So the one larva creates a bladder pack and then it starts budding out these daughter cysts. So that's how you end up getting something like what this poor female has on her face. So because of this kind of obvious nature of the disease manifestation, we were able to look at, I think at that point it was like 15 years of data. And we saw that, as we would expect from a parasite that requires the death of its intermediate host, of its larval host, to complete its transmission cycle, we saw an enormous impact on mortality. So for a female who was about 5 years old, who had a cyst, she had a 5 times about greater risk of death than a 5-year-old female without a cyst. And another reason why, so this is the mortality part, right? That's the one layer. But then we're really interested in looking at that second layer, right? What does the parasite do inside a single host that might be that endocrine manipulation that we were seeing in the Taenia crassoceps model? And it's interesting, particularly interesting to ask this in geladas because of their social structure. A little background on there. It'll be very brief, I promise. For male geladas, as in many animals, their fitness really depends on their competitive ability. So when you go to the Ethiopian highlands, you will often come across several hundred geladas all hanging out together on the plateaus. That's what we call the band, and the band is actually made up of a bunch of small reproductive units. So the reproductive unit is a bunch of related females and one leader male who has a limited tenure as the leader in that group. He has near exclusive uh, reproductive access to females. So he'll mate with all the sexually reproductive females in the unit, and then those offspring, when they reach sexual maturity, if they're females, they'll stay in the group, and if they're males, they'll disperse to what we call bachelor groups, which are just they're small groups of males that affiliate together, and then those males, as they become more mature, ready to start a family, they will challenge the leader males, not from their natal group, obviously, from different groups. And in that battle, if they win in a takeover, they become the new leader male. And what I want to emphasize about this is that it's really when we look at our data, when we look at the past 20 years, very infrequently do we see overthrown leaders start a second, successfully take over another unit. So it's really this discrete window of reproductive access. So they pour everything they have into this because this may be their only opportunity. And so obtaining tenure, successfully overtaking another male, and then maintaining that tenure is forms the basis of their reproductive success. So that means a high testosterone phenotype, one could say. And we see that leader males tend to have higher testosterone throughout the year. And then when the bachelors, there's a season where takeovers are more common, and the bachelors will then have higher, their testosterone goes up during those seasons as they start trying to overtake these males. So we used a combination of that longitudinal data and those and some cross-sectional data to ask how infection impacts sex hormones, with the hypothesis that Taenia serialis, much like Taenia crassyceps, manipulates the male endocrine environment for its benefit. So while most of our work is non-invasive, we, for 3 years, collected plasma samples during darting. So we would briefly dart the animals one by one and collect some plasma. So in the plasma samples, we assayed plasma testosterone and plasma estradiol, and none of these were repeated individuals. Individuals are only darted once, and those were done in the lab of Ben Trumbull here in the Center for Evolutionary Medicine at ASU. Then over our long-term non-invasive data collection, we collected fecal samples and assayed fecal androgen metabolites, which that's a little messier of a metric, which is why it was exciting to get to assay the plasma specifically. But each of these has, they're the pros and the cons. So we're much more limited in our plasma data set, but the fecal androgen metabolite data set potentially does not capture actual variation in what we want to measure, but it can be a proxy. And then for infection, we measured Taenia antigen. So I developed an assay to measure Taenia antigen with the Centers for Disease Control in 2015 or something like that. So we we looked for the Taenia antigen in the plasma samples, also cross-sectional, and then over a longer period of time, we have a urinary assay that we can use to non-invasively diagnose those. And then we also have the observation of cysts. So using the urine samples allows us to detect any cysts that may be not visible, maybe internal around the organs, or maybe obscured by their fabulous manes. And what we found when we looked at the impact of infection on estradiol was that infected males had 65% higher estradiol than non-infected males. So this is a huge impact. And this diverges from the sickness phenotype that we would expect if this were not manipulation, if this were just the body's physiological response to any kind of injury, any kind of infection. The body can divert energetic investment away from sex hormones into the immune response. And in this case, we're seeing this huge and significant impact of infection on elevated testosterone. So this really lends support to the manipulation phenotype that we also observe in the lab for Taenia crassyceps and indicates that that is happening here. But we looked at testosterone, and this was super surprising. We expected depleted testosterone because that's what we see in the lab. But here, there was no change in testosterone, which is pretty interesting, and I don't really know what's going on. However, we don't dart the leader males when we dart because we want to minimize the impact that we have on the reproductive fitness of the animals that we study. So this was just the bachelor males and then former leaders who are now followers, right? So none of these had that position where we would expect to see the highest range of testosterone. So that might limit our ability to detect anything. But then we turn to these fecal androgen metabolites, and because these are non-invasive, we were able to look at these in liters. And what we found was that there was actually increased and significantly increased fecal androgen metabolites in those leader males, in infected leader males. So this could, again, this is not necessarily reflective of testosterone in a really precise way. But one potential, so that could be one potential explanation. Another is that because of the importance of this window of reproductive access of male fitness, perhaps there is male compensation in the production as the testosterone is getting aromatized into estradiol. Perhaps that is triggering compensatory testosterone production as well. So then we wanted to look at how infection affected reproductive success because that's important because we wanted to. And our hypothesis was that Taenia serialis inhibits male competitive ability such that they lose their tenures. They're more likely to lose their tenures and have lower reproductive success. So for this, we turned back to that long-term data. We looked at the reproductive data for it was 96 males from 2006 to 2023, representing 103 unique tenures and 3,067 tenure months. And then we paired those reproductive data with infection data. So that was all the non-invasive stuff. It was the urinary antigen assay and the cyst observance. So we had 175 positive months and 19 males represented. And what we found will shock you. We found a really crazy impact of infection on on tenure length, on the risk of losing tenure in geladas. So here, this is the survival curve. On the y-axis is the survival probability, so the probability that tenure is retained. And males that are infected are in the green, and males that are non-infected are in the blue. And this is over time in months. And you can see that males that are infected almost immediately drop down the probability that they retain tenure that month plummets. And this translates in our Cox proportional hazard model into about 2.51 hazard ratio, which is about 151% higher risk of losing tenure. So this is a really, really substantial effect. And again, because losing tenure as that leader male means for many males that you're losing everything that you have. You're losing your ability to reproduce. So we use to kind of quantify the impact that that would have on males based on our the parameters in our population. We took the average share of paternity, we took the average females per group and the average interbirth interval, and we looked at how that would translate into loss in expected offspring sired. So what we found was that over the average tenure length, the infected males would lose, based on the number of months that they lost by that time, would lose approximately 1.5 offspring, which is also an enormous impact. So what we've shown is that Taenia serialis infection is really changing that endocrine environment in the hosts, and that may translate directly into this loss in reproductive fitness. It's also possible that there are other things. I mean, there are other things going on. This is obviously an extremely complicated system, and there's a lot of room to explore different mechanisms and to figure out exactly what that interaction with the immune system is going to look like, and of the energetics of infection, and how these are all going to kind of come together to produce this big impact on reproductive success that we're seeing. But this is our very exciting starting point. So, as a summary, when we're thinking about to bring it back to parasite manipulation, we're thinking about behavioral changes that precipitate host death as kind of the most the crux of what parasite manipulation is. But then what we're contributing to is this growing appreciation for parasite manipulation that can affect sex hormones that can affect the endocrine environment in ways that may impact host reproductive success. So this is also the starting point for my PhD student, Madhavi Verma. Her dissertation is going to get into these questions a bit more, along with a master student, Maya Bugos. So they together are going to be looking at genetic mechanisms, immunological mechanisms, and the behavioral mechanisms. So how does this translate, right? How does this change in their endocrine environment translate into the actual behavioral phenotype? So that's all I have for you today, and I'll take a second to thank you guys for being here and for the questions and thoughts that I anticipate being fabulous. And also to the Center of Evolution Medicine here and Club EvMed and all of our collaborators and funders. And so now I'm done.
All right. Thank you. Thank you, India. That was great. Amazing pictures as always.
Thank you.
Okay, we have time for questions. Does anyone, I think I'll suggest you stop the screen sharing, India, just so we can
Sure.
see everyone. And always nice if folks can be on camera if you're willing to. And let's see. I know we have a question in the chat from Neil. Neil, do you want to ask your question?
Sure. So I'm interested in whether you can rule out effects independent of the testosterone or estrogen levels in subverting leadership and loss of offspring success.
No, that's a yes, great question. Sorry, go on. I feel like
Well, the related point I was going to make is that I remember reading something by Robert Sapolsky about the fact that everyone assumes that testosterone causes aggression in males as the level increases. But what he was arguing was that testosterone, there's a complex interaction between other factors that it makes aggression more probable, but it doesn't actually cause it directly. And I'm just wondering if there are similarly complex effects of these sex hormones in terms of what it takes for a particular gelada to achieve leadership or to gain tenure, as it were.
yeah, I think that's a great point and definitely true. And it is certainly reductive to think of testosterone as a one-to-one, where you have 1 unit of testosterone is 1 unit of aggression, you know? Um, we are it, we do see when we look at testosterone in males across the year, we do see leaders generally have higher testosterone, indicating, well, I don't know what that indicates. Does it indicate that that they become higher testosterone, they have higher levels when they become leaders, or are the bachelors that have higher testosterone baseline more likely to become leaders? We don't really have an idea of that causality yet. But we do see also that the bachelor males during that takeover season, their testosterone goes up. So we know that it is these are related phenomena. But I think it's a great point, and it is hopefully what we're going to be able to get into is kind of taking this maybe simplistic uh, uh, formulation and kind of digging farther into what exactly is going on. So that's when we I'm really excited about the behavior stuff because we're going to be able to see are there behavioral changes. I'm hoping we can do some energetics too, but I haven't figured out how to, but I think that would be really cool. Um, and thankfully, we have all of these lines of data that we can start integrating to kind of get at those mechanisms.
Just another quick point is that I think there's some evidence now that changes in immune cell function can influence behavior. So there could be a more indirect pathway than just testosterone aggression or testosterone leadership behavior.
Absolutely. And you know what else affects immune cells, which affect behavior? Things like estrogens,
Right.
right? So we have this, I think, and this is, I mean, this is one of this is one of the frustrating but great things about working in the wild is that we can't get at everything. We only can work with the data that are available to us, but we can, I think that is a great point. And as we start, Medavi is also working on the immune question. And as we start understanding the immune response to Taenia serialis a bit more, that will shed light on some of those interactions.
Thanks.
Okay, thank you, Neil. And next, let's hear from Susan Alberts. Go ahead, Susan.
Hi,
Hey,
Susan.
nice talk. Thank you. And great, interesting system. Just really simple questions. I'm curious about the infection rate in the population.
Mm.
How prevalent is infection? And also, uh, since it's a predator-prey system, can you just remind us what the predators are and how frequently, how many of the animals in your population do you think are killed by those predators? I totally understand if you don't know, because we don't know for our population.
I love to think about that. Thank you for allowing me to talk about that. So, the it's a bit hard to say because we have this system where the cysts are really big, so we can look at our long-term data and we can look at, point prevalence or annual prevalence based on that. But we also know based on the urine data that infection may be more prevalent than you would think by just measuring the cyst prevalence. But it tends to range, and again, because animals die, like it changes all the time, but it'll range between 5 and 15%. And it is so okay. Well, I'll come back to that. But for the predators, we're working on that too. And we think there are several candidate definitive hosts. The jackal, there's two species of jackal. We've got the Ethiopian wolf and we've got the domestic dog. It's quite possible we just got all those fecal samples in like 2 weeks ago. There's been a little bit of war in Ethiopia that has delayed some stuff, in addition to all the other horrible stuff it's done. But we just got those fecal samples in, so we're close to finding out who the definitive host is. I would imagine it could be more than one. But also based on, there's that other gelada site, the Guasa gelada site, run by Peter Fashing and Nia, and they have a much higher prevalence of the cysts. And when you look within the simians, it's the northern side of the park that has the kind of higher altitude side of the park that has higher prevalence, and that is the range of the Ethiopian wolf. So, and Ethiopian wolves don't prey on geladas. In fact, at Guasa, they are fairly tightly tied. So, one thing is possible that when you just have a higher richness of predators, you just have a higher density of eggs being shed in the environment, regardless of who's eating you. Um, and predation. So, for the predation, we rarely see a predator event, but we have. That's how we got that one cyst was a leopard. And but what we do see, and this is what, again, one of the behavioral questions that I'm interested in, is that I was with one of these females that had this massive side cyst, Muppet, and she had her baby with her, and she was just it was impeding her movement so much that the rest of the band was half a kilometer away. And for geladas, that really puts them in a position. Separating them from the band is separating them from what we think is a predator avoidance strategy of that big group living. And the last picture I have of her, she's that half kilometer away, and then 3 days later, she was gone. So whether it separates them or directly kills them and then leaves them open for scavenging, both of those I think can play a role. Thanks, Susan. Caroline.
We're going to come to Caroline in just a moment. Let's go to Tom Strusaker, who posted a
Tom.
question. Tom, if you're there, are you willing to ask your question?
Hi, Tom.
Sure. How are you? Good to hear you talk. Marvelous stuff.
Thanks,
I
Tom.
just had a couple of questions here on so this reduced reproduction by the males, in general, how would that affect the female reproduction? Okay, there's 1.5 fewer kids produced, but in turn, would that be reflected in any way in population dynamics in terms of growth or reduction or stabilization?
That's a great question. Um, I don't know because there's never the males are never there's never more opportunities than there are males. So the position gets filled kind of immediately when a male is, or it does necessarily get taken over. So it's a replacement that I'm not sure. It's a good question. I don't know.
Oh, maybe we could couch it another way. The infected males have a shorter tenure in the group, band, harem, whatever you want to call it.
Well,
Which
it's
case
also,
it
oh,
may
go
not.
on.
I was just going to say, if it's a faster turnover with males who are infected, then it may not have much effect at all on overall population dynamics.
Yeah, and one thing that's important to note from the model is that what we're measuring is the likelihood of risk. And I think this is a really interesting. I mean, I'll be interested to hear what you think about the impact on population dynamics. But the model shows an increase in risk of getting taken over um, for infected males, but that doesn't necessarily mean a shorter tenure. I think I maybe said shorter tenure, but I didn't mean it. Because you can have a male that has been a leader for 6 years, and he's already enjoyed his reproductive access, he's had offspring, and then he gets a cyst, he gets infected, and then his risk of getting taken over goes way up. So, I mean, when we actually look at the individual data points, we see that that has happened a few times. It is often that it is much shorter than that. But there are a few cases, a few males that I can think of off the top of my head that already had all the offspring they could ever dream of. So in that case, that risk of tenure doesn't translate directly into the loss of offspring.
You can see this as being really a complicated question when you look at the age of the females and everything else and how their repro might decline. Just um, sort of a second question, if I may. That concerns that definitive host. Well, it's hard for me to imagine a jackal taking on a male gelada. I don't think it would last very long, and the jalaka would be consumed, but at least killed. I was thinking though that leopard. I remember when I was out visiting you, there was that leopard down on the face of the cliff. And the boons, the geladas, sorry, were very much interested, and I
That
would
was
think
awesome.
that's probably your guy. It's
It's not in felids. So it's
ah,
only carried by canids. And I that was one of my all-time favorite moments. Tom came to visit me. I think it was maybe my first or second time in the field. And we saw a leopard because the gelada saw it first and they all ran over to the edge of the cliff and started barking and then we ran over. So I think so the leopard won't carry it, but um
but you found it.
again it
You found
doesn't
a spore in the
necessarily have.
feces of a leopard, did you? Did you find some cysts or something in the feces of a leopard?
No, no, but
Okay.
they've never, I mean, I guess it's not impossible, but generally
I thought
there's
it
only
was.
records of Taeniid eggs in canids.
Mhm.
But it doesn't necessarily, again, have to be predation. It can also be scavenging.
Yes.
So I think that is more likely when we get to when we're talking about the canids, that it's killing the geladas and uh and also that the males don't have to be I mean, the parasite isn't perfect and it can it infects males, but it can also infect younger individuals that might be more vulnerable or females. um and I wonder, yeah, so that also an open question.
Right. Right. Yep. And always the classic old question: how do you distinguish between a parasite and a disease? Okay.
uh
Think about it. It's all right. But I mean, in this case, you're talking about, especially with those insects, that looks like a disease to me. Anyway,
Yeah.
thank you
I
very
would
much.
call, I mean, yeah, we can operationally define parasite. Parasite is a way of life.
It's a,
Um,
it's a trivial question.
but a fun one.
Okay.
Thank you very
Okay,
much.
I want to go on
Thanks,
to
Tom.
Caroline because her arm is getting tired with it held up
Caroline.
so long. So go ahead, Caroline. Good to see you.
Hi, India.
Hi,
Great talk. I
Caroline.
really liked it. I think
Thanks.
this might be along the lines of Tom's question, which is basically like, how do you think about the distinction between manipulation and like a symptom or just a consequence? And I'm sure you've thought about this more than I have, but
It's a great question.
yeah.
And it's yeah, that's it. So okay, so Robert Poulin had this kind of this big paper where he was like parasite manipulation, if you're gonna call it parasite manipulation, it has to meet this and this and this, and he outlined like 10 requirements, and then 10 years later he wrote an update and was like, I didn't really mean all that, like I think that really the threshold could be a lot lower. um and but I think it's, you know, we define these things, and generally what we're the operational definition that we are using here is a stereotypic change. So it's not just a one-off, and it deviates from expectations based on what we know about physiology or behavior. So in the case of the horsehair worm, it deviates. The mantises don't ever want to go into water. And then reliably, time after time, infected mantises will jump into water. So, and then in our case, we have our expectations based on what we know about sickness behavior and what tends to happen to sex hormones. And then this kind of reliable deviation from that is that in a way that, which I don't know, if we didn't know the experimental, have the in vitro data showing that it has a benefit for the parasite, I'm not sure that we would just jump to manipulation. But because it serves the fitness of the parasite, that's what that's what we think is manipulation. Thanks, Caroline. It's nice to see you.
Okay, let's Ned, are you still on? Would you like to ask your question? Interesting question in the chat from there we go.
Beautiful talk, beautiful system. Thank you so much. My question was about the red patch and if you see any alteration in the infected animals versus uninfected, and if there is an effect and there's a reduction in the red patch in a leader, does that make him more approachable by bachelors to challenge him?
That is such a great question. We are in the middle of our chest patch foray. Um, the it's hard because I have a really great undergraduate working on this right now for her honors thesis, Tiffany Duong, who was looking at those hormone data and the chest patch, right? Because I think that because that's what makes sense that you would that could be that mediator of if estradiol, if that conversion, if that aromatization has an impact on the redness of the chest, then you might expect to see well, actually, with estradiol, you might expect to see higher redness, but with energetics, with the energetic demands of the parasite through the immune response, you might expect to see a reduction in redness. And we're just not seeing any relationship, which is so puzzling. But also, again, we don't dart leaders. And so I think that really limits our ability to ask those questions about the chest patch, because the males that we have in our relatively small sample, which hopefully we can now start getting more, they're of a whole range of ages. Some of them are unknown to us, so we don't know if they're former leaders, which might have a different investment in the chest patch than bachelors. So it's a great question, and I'm hoping that we can shed some light on that one day because it seems like that would make a lot of sense.
Thank you.
Okay, thank you for that question, Ned. And I want to move on to another one in the chat. Ranak, are you on still? Hope I'm pronouncing your name right, Ronald.
Hi. Yes,
Oh, good.
Ron.
Fantastic.
Perfect. Thank you so much for that talk. Really, really exciting. One thing I was just curious about is whether you have information on the location of the cysts in these animals, perhaps also the size, in terms of determining amongst those who have the cysts, whether there's any differential effects related to the location of them. Because I could imagine that being a factor, but I'm just curious if you see that in the data.
I would love that. We have some kind of more qualitative data on cyst location and size, but it's basically because we weren't systematically collecting data on these at first, the data are more like giant horrible cyst on right face, which doesn't give us a good idea. But it's something that we will be collecting data on because I think it's also like it's important for understanding the impact on mortality, but it's also potentially important for this relationship with reproductive fitness and the relationship with the hormones, because you might expect to see more cyst equals more aromatase production equals more conversion to estradiol. You might expect to see that size of the cyst be or the location, like is it in a place that impinges movement? Is it in a place that obscures the chest patch? These are all, I think, really, really important. And but we have cysts that we can't just see. They're also inside. So I got a portable ultrasound machine for our last darting session. But then when I got there, I realized you actually have to know how to use it. So I did not successfully image a cyst. But in the future, that's possible, although we might have to shave them all. I don't know. That's neither here nor there.
Okay, I had one last question if there's time. I hope there is. When you were talking about parasite manipulation at the start of your talk, India, I was thinking, okay, evolution of virulence, it makes sense that where you have trophic transmission like this, and that's often where this parasite manipulation happens, you'd have highly virulent parasites and pathogens. And I thought, okay, there must be, there are probably going to be a lot of evolutionary counter strategies that will evolve then to avoid this. But then it occurred to me, well, if these effects occur late enough in life, in the life of the host, there may not be any selection against them, right? Because of the selection shadow and the declining force of selection with the age of the organism. And then right as I was thinking through that, you showed the mouse example with the increase. At first it doesn't have much of an attraction to the cat, but then it actually goes up. I was wondering what the units are and if you have any thoughts about that, if anybody's ever considered that, whether in essence, could it be possible that parasites make use of the selection shadow, right, to avoid, obviously, as a product of natural selection, to avoid, counter strategies by the host?
Yeah, I think that's true. That figure was infection progression over like 3 weeks of the rodent being
Okay.
infected with toxo. But I think that that's really possible, especially when you take into account that for geladas, when they're encountering eggs in the environment, and their likelihood only compounds of encountering eggs throughout their life. So that compounded exposure might mean that we have infections that are starting later on in life. And the same thing for what Tom and I were just talking about with the reproductive. You would imagine that if there's this huge impact on male reproductive success, you should see some
Yeah.
counter strategies. There's a lot of reasons why maybe you wouldn't. Maybe we're observing kind of a relatively new relationship. But also, there are those males that are still able to have already had all of their reproductive success and get that infection when they've already reproduced. So you would see selection shadow working on in that case. But I think yeah, it's cool because you look at it and you're like, this shouldn't be. There should be some sort of selection against that would save the host. But Not necessarily.
Not if it happens late enough in life.
Yeah.
Okay, well, thank you for a great talk, India. Great to, okay. Oh, yes, Joe is, I'm glad you're here, Joe. I want to announce the Evolutionary Medicine Summer Institute that TriSem is hosting. I've just put a link in the chat there. It's focused on climate change and health. Those are the dates that we have for it. And this is also done in collaboration with some of the other centers, including the Center for Evolutionary Medicine, which is, of course, India's affiliation, and the Vanderbilt Evolutionary Studies Initiative as well. So anyhow, please consider applying and coming to that, applying to it. You can find more information on the link. Great talk, India. Fantastic.
Thanks for
Thank you for
having
joining
me.
us. And
Nice to see everybody.
hope to see you in person soon.